Good afternoon. We are starting the conference of the International Genetic Federation, and it's a great pleasure to me to present you Professor Amilcar Tanuri. Professor Amilcar Tanuri is full professor of the Federal University of Rio de Janeiro. He is graduated in medicine by the Federal University of Rio de Janeiro, where he also got his master and PhD. Currently, he is consultor of the World Health Organization, coordinator of and consultor of several agencies in Brazil. And he is associate research scientist in Columbia University. And he is one of the greatest Brazilian references in the virology. Over the years, he has mainly studied HIV, its genetic diversity, and virus drug resistance. Currently, he is studying COVID-19 pandemic, seeking to understand the immune response to the infection. Amilcar, it's really a great pleasure to me to have you here, and you have accepted our invitation. Thank you very much, and it'd be really a great pleasure to hear your talk. Thank you.
Okay, Marcia, thank you very much for the presentation and introduction. And I will start my talk. Today we're going to talk about the COVID pandemic here in Brazil and the world. This is where it all began. This is the fish market in Wuhan, where the case start to be fine, but nobody knows if this is actually the center of the spread, or the virus came to here from with someone infected and spread. It's more likely that the virus actually was selected outside Wuhan, and someone came here and start spreading the virus. Is much more likely that the virus actually is being selected in human beings for a long time till it becomes actually able to spread human man to man and actually spread the virus all over. This is the normal way we get actually the beta coronavirus, mainly beta coronavirus from bats, but also you can have from actually mice, and then you have intermediary animal, and then goes to man. So this is the route of the transmission and the zoonotic transmission from bats to man. So we have the old SARS-CoV, the MERS, and now in the new SARS-CoV-2. In the SARS-CoV-2, we don't know really what is the intermediary animal so far. People think that is pangolin, but can be other animals. But this is still to be clear. In a near future, a lot of research will be doing to know what is the intermediary animal of this virus. So this is the picture of our enemy. It's a coronavirus, and you must know that coronavirus is a RNA virus, and it has the biggest genome among the human RNA virus. It has 30,000 nucleotides, around 30,000 nucleotides, and is the virion is actually decorated with a spike protein that is the major protein that we drive our neutralizing antibodies, and is actually the protein that actually makes the interaction with the cell receptor and puts the virus inside the cell. So This is the structure. You have here besides the spike protein, you have the envelope protein and the matrix protein. Here is a cell not infected, and this is the cell infected by SARS-CoV-2. And you have this phenotype or the cytopathic effect that is mainly the formation of syncytium. And also this is the electron microscopy showing the formation of the virion inside these vesicles we call viral factory, these vesicles where the assembly of the virus is done. And this is the vesicles going to the outside the cell. Here is briefly the genome structure of this coronavirus. So this is the structure of SARS-CoV, MERS. The SARS-CoV-2 has very similar structure compared to this virus here. So the main thing is that you have two ORFs, the big ones, and then after you have the structural proteins on the right end of the genome. In the left end, you have the ORF1A and 1B, and they are responsible to produce a lot of proteins that give rise to the replication enzymes of the virus, mainly the enzymes that makes the genomic RNA in a complementary RNA, and also produce the new mRNA that will be translated, and also a lot of subgenomic RNA that also will give rise to the structural proteins like spike, envelope, membrane, and nucleocapsid. Besides this protein, you have some extra proteins here, open reading frames, that are also important for the interaction between the virus and the cell. Of note, the 7A and the 8A peptides, they are important to deal with the interferon response of the cell, and they are likely to block the interferon response and makes the virus more able to replicate inside the epithelial cells to make its pathogenic effect. So this is the cycle of the virus inside the cell. So you have the entrance. You can have a direct entrance through the receptor. The receptor of SARS-CoV-2 is the receptor ACE2 that belongs to the pressure response, is an angiotensin receptor, and the virus can be have a pinocytose and also and release the genetic material inside the small vesicles, and the RNA will be released. The RNA is a positive strain, and you have the production of the viral protein, including the replicase. Replicase will make the complementary RNA, and also a series of subgenomic RNA that will be translated to the viral proteins, and the genomic negative strain gives rise to genomic positive strain that will be packed, and then you have the release outside the cell. So for the viral entrance, you need the ACE receptor and also a protease that is a cellular protease that will cleave the spike protein between subunit 1 and 2, and this cleavage accelerates the fusion of the membrane, and then you have the release of the protein. A lot of people are trying to inhibit this protease, the cellular protease, trying to block the entrance of the virus. But many studies are doing now, are trying to develop a specific antiviral to SARS-CoV-2, but so far we are not succeed. Actually, the only molecules that we are finding are some inhibitors of the RNA polymerase of the virus. So we have two molecules that we went to clinical trials, remdesivir and also a new one from Merck. But both of them are nucleoside analogs that actually inhibit the RNA polymerase. So this is the interaction between a small piece of the spike protein called RDB with the ACE2 protein. So when you have this interaction, there is a change in the shape of the spike protein, and then you have the cleavage of the spike protein by the cell enzyme, and then you have the fusion of the membrane. So here are the entrance key and locks. So this is the ACE, the structure of the ACE2. This is the structure of the protease, the cellular protease. And they are very key elements in the spread of the virus. So here is the beta coronavirus tree showing that SARS-CoV-2 is a completely new virus, and this virus has not came from any of the old SARS-CoV we know. That was the SARS-CoV-1 and the MERS. So this is a brand new virus that was selected in nature. So a lot of people claim that this virus can be made in the test tube, but is actually only speculation and is not true. So COVID-19 epidemiology in Brazil and Latin America. Unfortunately, Brazil is one of the major targets for COVID-19. Here is the statistic done in September 26. So Brazil got like, now we have more than 22 million cases reported and nearly 600,000 deaths. So with a mortality rate very high. Brazil, this is the curve of daily cases in Brazil. You see that we start our pandemic very, the first case was diagnosed in March 2020. And then we got a first wave that started in April, May, and we got a huge number of cases by July 2020. And then the number of cases went down, and then we got a second wave by the end of 20, like November, December, and this wave lasts for a long time till July 21, when we have a sharp decline of the case. And we're gonna talk about that. This phenomenon here is more likely to be related to the vaccines. This is the death. It is a curve that follows the number of cases. And you see here a steep decline in the deaths after July. And what is interesting is that this decline in the deaths goes with the is actually coincident with the increase of the vaccination in the country. Right now, by 23 of September, we got nearly 150 million individuals receiving at least one dose, and at least nearly 70 million receiving the full dose, this 2 doses or the full vaccination. So you see that with the increase of the vaccination, there is a decrease in the deaths. So we are very happy with the result of the vaccination here in Brazil. And it makes us more comfortable to tackle the epidemic because you're gonna see that in the further in my presentation that we face two different variants. In the beginning of 21, there is an increase in the number of cases actually driven by variant Gamma, and variant Delta is starting in Brazil in July, but as we have a lot of vaccination, Delta established but never increase the number of deaths or increase the number of infection. So it seems that the vaccine actually blocks Delta very well here in Brazil. It's amazing because in Israel and United States, you can see Delta growing even in people vaccinated, but you know, in terms of infection, not deaths. But in Brazil, we have a hit, the vaccine actually block even the infection with Delta. So it's an interesting chapter to study. So, so far, in a recent data, we have 230 million doses apply, and we have a total of 40% of the population totally vaccinated against COVID. Okay, the first problem we got in the beginning of the pandemic was how to diagnose well the case. So this is the curve of the diagnosis. So here is the RNA when you are exposed, and then you have acute phase, and you have the symptom onset here. You know, the incubation period of the virus can vary from 2 days to even 10 days. But when you start having symptoms, you increase the amount of virus in your nose secretion. And then you have also the antigen of the virus increasing. So both RNA, PCR, or antigen tests are very good to define the acute infection. And after 15 days, you start having the IgM, and then you start increasing the IgG. So this is the curve of people primarily infected. So you have this kind of shape. But you see that now all these things change with the vaccination and also people that got reinfection. So this, for example, the antibodies can have problems and to be used because of this kind of events. Hang on one second. Esse barulho daqui é de fora. I have a noise here at the lab, sorry. So one test that was used a lot during the outbreak was actually the rapid antibody test that can actually measure IgM and IgG. So this was a very useful test at the beginning to show people that was infected in the past. There was a big problem in Brazil and all over the place. We started using this test to isolate people, and was a mistake because when you have the antibody, actually the infection has gone, and actually the phase that you are more infectious is gone. So you are much more infectious this phase than this phase. So the control of the spread of the virus needs to be done with PCR, but now we have a very good tool that is the antigen test. So the antigen test is the best test to be used when you want to isolate and identify people infectious and actually spreading the virus, and the people that you need to actually to do the isolation. So this is a work done in my university together with another colleague from the biotech department, COP, where this collaborator, Leda Castillo, expressed the spike protein in HEC cells, in mammalian cells, and purify the spike protein here. And then we used this protein to do a ELISA test. So we developed a ELISA with trimeric S protein. And you see that this trimeric protein is very, very sensitive. And it has a window after 10 days after infection or symptom onset. You have nearly 15 days, you have 100% of positivity in the ELISA of people infected. So this is the curve of persons with a PCR positive at the beginning. Then we did the follow-up, and you see that the test is very, very sensitive. However, if you start following up the patients for the same patient all over the time, you see that the antibody against the total S protein stays stable for at least 110 days. But with the RDB or the S1 structure, the antibody goes down and you have a washout, and also in the nucleocapsid. So, so there is a kind of weaning of the antibody when you go for long time after infection. So this is a concern, and actually this can make the individual after 4 months after the infection susceptible to another infection. And this is very common in the biology of coronavirus, the old coronavirus, the normal one. You can see people that can be infected with the same coronavirus strain many times during the life. So this is a virus that actually can do reinfections. Here is another phenomenon that we saw in our studies, is that a rapid test that measure antibody against RDB, you can have like a seroreversion of the test as soon as the infection goes in a long time. See here, individual that 4 days after infection, there is no reaction, 22 days, the reaction starts with the antibodies identified, but with 182 days, the antibody is washed out. So the same for this patient retain, but this patient also lose the antibody production. And this is the curve we saw using the MED test and the wonderful test, and you see the phenomenon of seroreversion. So another kind of test that show up during the pandemic last year in the market was the antigen rapid test. And this is a very good test because actually measure this part of the curve. Actually, the antigen test identify the virus itself in your swab, in your nasopharyngeal and nasal secretion. And this is a very good test, and it can be done with immunocapillarity. So basically, the test actually has a monoclonal antibody attached to the nitrocellulose that capture the antigen, and you develop the antigen with an antibody against the antigen linked with a color. So you have a band, very easy that you can identify. So here in my university, we evaluate PanBio test. That is an antigen test, very good, that we could actually identify. It's easy to perform, like in 20 minutes, you can run the test. Here is the site we organize our testing in my university. So the patients arrive and they have an interview, then they collect the swab, then they do the antigen test and also the PCR. And we could actually study a lot of the immunological response of the virus during all this pandemic. And also we could organize a sera bank of individuals after infection and also after vaccination. So just at a glance, I'll show one of our study that we evaluate the performance of the antigen test. So we collect samples of individuals with less than 7 days after symptom onset. And then we run the PanBio antigen test, the antibody test, and the PCR, and then we analyze the performance. So you see here the result of one of the study showing a very good sensitivity of the antigen test compared to the PCR gold standard. So you have, PanBio has like 91.4% of sensitivity, meaning that all the individuals infected are in the 91% of the individual infected. They are identified by the PanBio. We miss part of them, but the specificity is very high. So all the negative individuals, we have very few false positives. So we are very happy with this kind of test. Also, we test this one with nasopharyngeal swab, and then we test with nasal swab, and the results are very comparable. So nasal and nasopharyngeal are very good. And here we perform, we compare the agreement between nasopharyngeal and nasal swab, and then the agreement is very, very high, it's 100%. So meaning that you can do the test both ways. So here it is the study 2 with a symptomatic patient. You see that the test actually can identify a symptomatic individual, the antigen test, but is less sensitive compared to the symptomatic individuals. So, still, if the individual asymptomatic has a high viral load, the test is very sensitive. The problems are the individuals asymptomatic with very low viral load in the secretion, the nasal and the nasopharyngeal secretion. So the test perform less well when you have a very low viral load. So this is a new evaluation we did that we are very happy, is the self-collected swab. We made the patients to collect their own swab and actually perform the test. So this kind of rapid test is able to actually perform very well with people doing themselves the test. And actually this was done by October 2020, and this time was very interesting. This result based the decision of WHO to promote the self-collection test. So showing that if you do the self-collection, the sensitivity doesn't change a lot. So showing that you are able to do the test. So anyone can do the test at home. I hope in Latin America, these antigen tests are released to be done at home and help actually moms and to send the kids to school, and many initiatives can be promoted, safe measure doing this test. Another thing that we observe is that if you have a positive antigen test, means that you are more infectious. So if you can identify individuals with antigen test positive, you actually identify more infectious individual that needs to be isolated. So it's a very good weapon to segregate individuals infected that can transmit the virus. So we believe that the use of antigen tests in large scale can make our human activities more safe, such as schools, events, and actually work. So let's talk about vaccine. So vaccine is our main intervention to combat COVID. Vaccine is one of the most effective ways to stay healthy and prevent infectious disease, especially respiratory disease. So it's a smart way to deliver antigen from pathogen to host without causing disease. That's the thing. So we have different platforms to develop to make vaccine. So you have a vaccine with attenuated or inactivated virus. So yellow fever is attenuated, for example, inactivate pathogen coronavac, and you have recombinant protein that you isolate and make into vaccine with a subunit, and you have VLPs also, and you have viral replicative, that is a virus that doesn't cause disease in man, but carries the antigen of COVID, for example. And you have a non-replicative virus, that is the AstraZeneca vaccine, that carries, is a disarmed adenovirus that carries the information inside that produces spike protein. And you have the crude DNA vaccine, and you have the mRNA, so famous, that start being used during this pandemic. So these are the vaccines around the world for COVID. So you have different kinds of vaccine. Actually, we have 99 vaccines in clinical development and 184 vaccines in preclinical. And actually, we from Latin America, we need desperate to have more countries trying to produce their own vaccine to have a kind of autonomy and not to depend on the vaccine produced by developed countries only. So this is a very sensitive issue here in Latin America, and I think we need to promote this kind of research in our universities. Here is a very interesting test we use to people after the in the patients after the vaccination. Is actually a test developed by Promega, and actually measures the binding between the RDB and the receptor ACE2, and is a very ingenious test where you have a bite. So you have the interaction between the ACE and the RDB, and when the two proteins interact, you have two pieces of monoclonals, each one carrying a piece of the luciferase. So each one separate, has no light. When you have the interaction between ACE and RDB, the two subunits or pieces of the luciferase actually match, and then you have the lights with the substrate. So this is a very, very sensitive way to measure the interaction between RDB and ACE. And if we add to this reaction the serum of the patient, if you have an antibody that blocks this interaction, this antibody is likely to be actually neutralizing antibody. Actually, our group worked together with the Promega group to validate this assay. And in our hands, the assay performed very well. For example, look here, the reaction limit after coronavac immunization. This is the production of IgG after immunization, first dose and second dose, and then you have the increase of the IgG, and here is the neutralization of the reaction here. So without neutralization, you have 100% of light, and then the light is disappearing here, okay? So this is a very interesting result, and this is also the humoral response of individual vaccinate that was previous infected. So you see, when you are infected and you have low titer of virus and neutralization, but as soon as you receive the first dose, if you are infected, you increase the production of antibody very fast, and also the neutralizing antibodies. So this shows dramatically what the vaccine does. The vaccine actually protects you two ways. If you are not infected and vaccinate, when you are infected after vaccination, you start producing antibodies very, very fast, and then you reach very high concentration of antibody in the blood, and also the neutralizing antibody that makes you makes you respond very well to the infection. So that's why the vaccine protects you against severe case of COVID. So this is the individuals vaccinated with 1 and 2nd dose of CoronaVac, and you see the decrease, and the neutralization goes the same way. So we actually could show that when you have the title of IgG high, you have the neutralization very high. So this is a very important test. We are promoting this test, neutralization test, LUMIT, to show the immunological status of vaccinated individual. So we think that not only the vaccinate people, actually the amount of neutralizing antibody is the good measure to see if you are protected or not. So this again, the LUMIT, and also how LUMIT behaves during the natural infection. So you have the antibody here. When you get infected, you increase the amount of antibody and also the neutralizing antibodies. And here is another interesting thing, natural infection after first dose of CoronaVac. So when you have CoronaVac, you have an increase very slow. And then when you are infected, you have a very sharp increase in the production of antibodies that is low here, and the neutralizing antibody, showing that the vaccination, even if you don't have a lot of antibodies, when you are infected, you start producing very fast the antibody and protects you against the severe form. And here is another interesting case where we could actually show differentiate people that are vaccinated against people that was vaccinated and infected. So here is the reaction of anti-spike and anti-nucleocapsid. You have to bear in mind that the vaccine has only the spike protein, not the nucleocapsid. So here, when you are immunized, you have the increase of the nucleocapsid, the spike protein antibody. Nucleocapsid stays in the bay, very low. But when you are infected, look, you have the increase of the IgG anti-spike and also anti-nucleocapsid. So if you run ELISA using anti-spike and anti-nucleocapsid, you measure both antibodies, you can distinguish between vaccination and infection. So this is an interesting tool to explore the cohort of people vaccinate to check for infection after vaccination. Okay, so let's go to another chart we do in our lab, is the study of the variants. This is a very important paper published. Our group helped also participate in this study, and we show of the infection in Brazil by March 2020 till May 2020 was the very beginning of the pandemic. And you see that in Rio and São Paulo, you see that the case increased very fast and the deaths. And here is the R0 rate, that's the reproductive rate, and was very high when we started the measure of isolation. So we have a decrease in the R0. However, in Brazil, due to the lack of really good isolation measure, this R0 never went down to 1. So always our R0 is above 1. Only right now, after vaccination, that we have sites in Brazil where the spread of the virus is very, very low. But this time, actually, the virus spread very fast. And actually, when you do the sequence, you see virus that came from abroad in blue, and actually, the autochthonous virus from Brazil are here in red. And you see in the phylogenetic tree that the autochthonous virus actually start differentiating in new variants, is this clade 1 and 2 that are actually the B13 and B1133 and B1128. And then you see the first wave was driven by virus imported, but only the second wave very fast start with autochthonous virus, and then you have here the curve of you see that when you block the transportation, international traffic, you decrease very fast the virus imported and not the virus autochthonous. So this is the spread inside Brazil. You see that at the beginning, the spread was located in the coast, but after May, the virus spread all over Brazil, and then you have the situation of very drastic infection. And here is the GSIDE site showing the different variants all over the world, showing that the Delta virus is taking over this year. And right now in September, Delta virus accounts worldwide more than 90% of the infections. Unfortunately, in Brazil, Delta arrived also in Rio de Janeiro. I will show you later. And this is a very important variant of concern. Here, our gamma, that is our P1. You see that still you have some numbers here, but actually Delta actually overtake all the variants here, and every variant disappeared. The Alpha was replaced by Delta all over the world too. So here is a very interesting time course of the Zeta VOI variant of interest called P2. P2 is a variant that derived from the B1128 strain, and so you have this clade here, the P2. And interesting that in May 2020, you have only the B1128 and 33, and then suddenly the P2 starts showing in October 20, and then it starts growing very fast in Rio. And then after December, P1 arrive in Rio and overtake P2. Right now, there is no P2 variants in Brazil. So this is the difference between P1 and P2. This is the phylogenetic analysis. They are very different virus, but both of them derive from B1128. So look the amount of mutation or SNPs, non-synonymous in the genome of P1 compared to P2. So in the spike, P1 has a lot of mutation, like 11, and P2 has only one mutation. So, and this virus is very fit and actually can transmit very fast. So this is what we see here in Brazil from May 2021, when you have the gamma strain overtaking the zeta. So here is the curve. You see here the show up of P1, and then P1 was taking over, and then you have the replacement between P2 and P1 in Rio. So P1 came from different parts of the country in Rio de Janeiro, like the Central East, North. So actually we got like 13 introduction of gamma strains in Rio. So it was a multiple introductions, interesting. And the replacement was very, very fast. P2 variant was replaced by P1 in less than one month. But the reason is here. You see that the amount of virus measured by the CT of the N1 target PCR in the P2 variant is higher than in the P1, meaning that P1 express or shades much more virus in the acute phase of infection compared to the P2. Then the individuals infected with P1 has a huge capacity to transmit the virus compared to P2. That's why P2 was overtaken by P1. So you see here that the average CT of the virus goes down during the increase of P1 in our cohort. So P1 actually has a different biology compared to P2. And recently now, Rio de Janeiro is amazing because we have replaced from P2 to P1. And recently, in July 2021, we start seeing Delta arriving in Rio in June. Delta was 6%. In July, 48%. In August, 89%, and right now is 95%, 99% are Delta in Rio. So you see that Delta actually was actually a very, very aggressive virus in terms of not disease, but actually spread. But there is a very interesting phenomenon. Although Delta replaced P1 in Rio, the mortality and the number of infection decrease. So that's why we think that this is a major effect of our vaccination. And this phenomenon needs to be more studied. Actually, so a lot of hypotheses can be made here. Maybe P1 variant protects against Delta in terms of severe disease, but not infection. Or P1 actually, the transmission of Delta is much more effective compared to P1. So it's something that we need to study more. We need to isolate these variants, P1 and Delta, and put them to culture and study some biological property of these two viruses. It's very interesting work to be done. This is another work we did with patients with prolonged infectious virus. So we collect like 33 patients that had positive PCR above 14 days of symptom onset. Actually, they got prolonged shedding of virus. So we try to study if this prolonged infection is driven by mutation in the virus. So here is the phylogenetic tree of individuals with, look, this is the first sample in purple, second sample in purple, first sample in orange, and you see that they belong to the same branch, most of them, so you don't have a very different virus. They are not different, they are not reinfection. These prolonged infected individuals is not done by reinfection, but actually through evolution of the infectious virus. And here you see we observe diversity of the virus as soon as the CT was going down. So what we saw is that at the beginning, obvious, of the infection, in this prolonged infection, the viral load was very high. Actually, the CT is low. But at the end of the prolonged infection, the CT is very high. The viral load is very low. But look at the genetic diversity. This increase all over the time. And what is interesting to show here is that this mutation accumulated in the long run, most of them are related to predicted T-cell epitopes, showing that this is driven by the immune selection. And we hypothesize that this is the way the virus actually or the new variants arrive. Maybe the new variants came from prolonged infection in human beings in the first wave of the infection. And then the variants are selected by the antibodies of these patients, and actually they overgrow later. Here is the mechanism, very interesting. So at the beginning of the infection, there is like increase in the A2U mutation, decrease, and you have a decrease in the G2A mutation, increase to G2A we observed two different mutation paths driven by increasing A2U and decreasing G2A. The G2A was associated with the RNA editing property of the Applebacks, and the A2U maybe something driven by the RNA polymerase. So you see here that in time 1, you have a huge change J2A, and when you have the time 2, this number decrease. So showing that there is some shift between G2A and then here you have the opposite. So maybe this is driven actually by the host innate immunity here, the enzymes that actually blocks the RNA replication. And here is the A2U that you have low in the first time and high in the second time. So we need to study these properties. Here is our group trying to study the difference between P1 and P2 in terms of neutralization. In this case, we use the serum of an immunized horse against SARS-CoV-2 protein, showing that this serum is less able to neutralize P1 variants compared to the P2, showing that maybe there is a difference in the neutralization profile of this virus. And so, challenge for Brazil in the long run, we need to control the transmission, vaccinate the population as quick as possible, and do also some boost in people with a low amount of antibody after the vaccination, like elder people and people with comorbidity, to do like a booster or a third dose, and then target specific population to give vaccine boosters, and also test more and isolate people infected. So here is another small project we have in our university. We are trying to develop our own mRNA vaccines. So we have a PhD student doing this work, and a big group of our students are actually involved in this protocol. So we are trying to produce our mRNAs, modify the mRNAs with and nucleotides modified, and then immunize mice and see if we can have the production of antibodies. We are very hopeful that we will be able to develop our own mRNA vaccine. Thank you very much, the financing agents, and this is the group of the lab and all the people involved in this study. Thank you very much for the patience. Sorry.
Amilcar. Yeah. Thank you very much, Amilcar. Thank you very much. Wonderful, wonderful talk, and wonderful. We have learned a lot with you this afternoon. And I think I would like to say congratulations for the development of new vaccine platforms. I think we are proud of our university doing so nice work.
Yeah, we are trying. Actually, it's very difficult right now to do it because a lot of reagents are lacking, and we are struggling to have the reagents here. And we are trying, but it's a challenge to have.
That's why it's still more really. We have really to say thank you and congratulate you and your group for this initiative. So I think you have a question I send you by WhatsApp.
Okay. Yes, Melania Abreu asked, the neutralizing antibodies miss with time. Do you recommend a booster dose? If so, in what case? Yes, what we found is that the titer of neutralizing antibodies goes down. With CoronaVac, goes down very fast. AstraZeneca less, and Pfizer also less fast. But all of them, you see a kind of winning in the amount of antibody that reverts when you do the booster dose. So, however, what we observe, I show you, is that if you are vaccinated and you have infection after vaccination, the amount of antibody produced is very, very fast. It's very high and very fast. So we were very hopeful that, and actually this is the reason why the vaccine protects you against severe forms of the disease. Is because you make more antibodies fast when you are vaccinated. But I agree with Melania, it's a very important way to I think it's a very wise doing like a booster dose in the elder people and people with comorbidity, because after 6 months, we observe a decline in the amount of neutralizing antibody in vaccinated people. That's the answer. We agree.
Okay. Thank you, Amilcar. Some people are thanking you and saying excellent conference. Thank you very much. But I have some questions. I think we have 2 minutes to discuss this.
Okay.
The first question is about if you could be confident that the virus was not produced in laboratory. You talk about
yeah. Yes, we if you analyze the sequence of SARS-CoV-2, you see that all the sequence evolved naturally, and there is no like cut and paste of sequence from different virus. If the virus were done in the lab, we tend to mix pieces of virus and make a new virus. And this you can identify when you analyze the RNA sequence of the virus. So I am in the group that we don't believe that the virus was done in the lab. The virus was done in nature that has a very strong way to select. So if this was selected was actually maybe in the countryside of Asia, in China, where the bat infected some animal that infected man, and the man infected back animal, and the animal back man, and this selection process leads to a very, very selected virus that can actually infect human beings and make this pandemic. So this is a very slow process. It's not done by chance. It's actually selected.
Okay, I still have another question. Indeed, I have a lot of questions, but I'm sure that you don't have time to discuss all of them. But I'd like to ask you how to explain that some people, some person has a kind of immunity. So, because you know that some examples of people have living together with other that got the coronavirus, and that person does not. So how to explain?
We have a professor here in our department, in our lab, that is studying this kind of different genetic makeup of individuals, especially in the genes related to the immune, natural immunity, and some individuals has alleles more prone to react against the SARS-CoV. So I think this kind of human variation can lead to actually this susceptibility or different susceptibility to the virus. I'm sure, but we need to discover.
Okay, so one more question from audience. Considering your experience, I didn't send you. Are you just reading?
Okay.
Considering your experience in this pandemic, could there be a third wave? This question is from Soledad Berrios.
I hope not, Soledad. I think the vaccination makes a kind of barrier against deaths in severe case. We can have a wave if a new variant arrives, but I don't think this will be new enough to make a more lethal virus. Actually, maybe the virus can spread, but not induce very severe case. But I think we proved that the best way to combat COVID-19 is the vaccination. I think this is actually well proven.
Amilcar, what about the immunization after infection?
Yeah, the immunization after infection works if you are infected after when you have negative PCR, like 14 days or 1 month later, you can be vaccinated, no problem. What happens is that you produce much more antibodies.
So I was thinking about that's
Yeah.
very efficient. So I think no more
Yeah.
questions. And you are time to finish. So thank you very much. I'm really very happy to.
No, my pleasure, and I'm very happy to participate in this meeting. And
learn a lot. Yeah.
Thank you very much.
Thank you. So thank you for audience, and I think we close this session. Thank you very much. Bye bye.
Thanks.